Hello အားလုံးပဲမင်္ဂလာပါ My Passion to Yours ကနေပြန်ပြီးတော့ကြိုဆိုလိုက်ပါတယ်ဆိုတော့ဒီတစ်ပတ်မှာကတော့အမ guest speaker တယောက်ကိုဖိတ်ထားတယ်ပေါ့နော်ဆိုတော့အမတို့ရဲ့ guest speaker လေးကိုတစ်ချက်ဖိတ်ခေါ်လိုက်ရအောင်ပြီးတော့သူဘာ topic လေးပြောမလဲဆိုတာကိုလည်းဒီကနေပဲတစ်ချက်မိကြမယ်ပေါ့နော် Do you know how to introduce in my language?
I can say hello name Mingalaba. If that works.
Yeah. Okay. So can you introduce yourself first?
Okay, so I am Juan. I'm currently a PhD student in Taiwan. And my main focus is in agrobacteria, which is what I'm going to talk about today.
Oh, okay. So what made you interested about agrobacteria?
So I would say the most interest about agro is that you can use this bacteria for doing plant transformation, which is what I'm going to explain today. And aside from that, it's also a plant pathogen. So studying plant pathogens in general is also good for many purposes, like for diagnosis, because you can imagine that plants also get sick. So it's good to know if how the disease happen and if we can treat or prevent that.
So let's start explaining fact by Juan.
So as I mentioned before, agrobacteria is a series of plant pathogens, and we call them soil-borne because these bacteria are naturally living in the soil. So when they are not, of course, they are not infecting the plants all the time. So these bacteria are very special because when they infect the plant, they produce a very obvious phenotype. So here we can divide the bacteria either in crown gall producing. They are called like that because when they infect the plant, they produce this undifferentiated mass of cell mass in the infection site. Or we categorize them as heavy root producing. And here you see a lot of roots coming from the plant, but actually these roots are not the natural roots from the plant. They are produced by the bacteria, and you can distinguish from normal roots because these ones have a lot of hair. That's why they are called hairy roots. So how does this bacteria produce either these two phenotypes? So here in this drawing in the left that you can see from our PhD student in our institute that is called UYU, you see that the bacteria has this small circle, okay, with a yellow line. So we call this oncogenic plasmid. These bacteria use this oncogenic plasmid for producing something called T4SS or type 4 secretion system that I might explain to you later in more details. And these bacteria use this type 4 secretion system to infect the plant cell. So this thing that you see here is a plant cell, and you can see that the bacteria is physically injecting the plant cell with the yellow line that I mentioned before. And this yellow line has a series of bacterial genes that when they are translocated to the plant cell, they are going to be expressed. And by expressing those genes from the bacteria, the plant is going to have that phenotype that corresponds to both the crown gall or the hairy roots, depending on the type of plasma.
Can I ask one question?
Mm-hmm.
So what type of, can I say the disease type here, or how should I refer to crown gall and hairy root?
Yeah, so this is pretty much the name of the disease. So crown gall, when you say crown gall, you always refer at this kind of like a tumor. And when you say hairy roots, you always refer to this type of root.
I see.
So actually that's the name of the disease.
So which one is more common caused by agrobacteria?
So both of them are common, but the most studied one is the crown gall because that was the first one to be identified. You can imagine that when like 50 or 60 years ago when this was discovered, people just look at the plant and saw something weird, that the plant normally doesn't look like this. That's why you can imagine the first one to be identified was the crown gall because it's more obvious than the hairy roots because they are underground, right?
So can it also depend on the type of plant that they infect?
So in terms of we call that host, type of host, and that really depends on the bacteria. Not all the bacteria has the same host, and there are different bacteria that produce both phenotypes in different hosts. And actually, that's an interesting question because that's not very well studied. That's something we don't really know about. People there are not really many people interested in that, and the reason for that is because the main interest of agro is for tDNA delivery and not because of the phenotype.
I see. Okay.
Okay. So, as I mentioned before, what is important about agro is that it can deliver tDNA. So, sorry, it can deliver DNA to the plant cell. So here, first I want to talk about the left side, and the DNA that is transferred from the bacteria to the plant is called transfer DNA or tDNA. They mean the same thing. And this tDNA is not transferred to the plant cell by itself. It's always accompanied by several other proteins, and these proteins, we call them effectors. So here we see the plasmid, the plasmid, the oncogenic plasmid that I talked about before. We can see it here in more details, and you can see that it's called PTI or PRI, depending on the kind of phenotype that you observe in the plant after it's infected. So here you see a red region, which is the tDNA region, which is the genes that are going to be expressed in the plant, and a blue part of the plasmid is called the BIR region, and this region encodes the effectors that are going to help to translocate the tDNA to the plant cell. So here you see that the tDNA, which is the red line, is removed from the plasmid and physically transferred to the plant cell. But you can see that that's always done together with different proteins, not by itself. Otherwise, it wouldn't be possible to translocate them. Okay. And the other part of the system that I want to, or I already mentioned, is the type 4 secretion system, which is the machinery that you see here. And you can see that machinery is very complex. I think that's a very cool part of agrobacteria, because this is telling you that how complicated are actually things. Because you can imagine, normally people in the lab just put the bacteria together with the plant and you observe the phenotype. But what is cool is that that's done in a very sophisticated and very complicated way that people not always realize how difficult it is to do that to the microscopic scale. Okay. So the last, of course, the last thing that has to happen is that the tDNA is moved from the bacteria to the plant cell and eventually integrated into the own plant genome. So all of this process happens during transformation, and well, let's stop here for now. So this is pretty much what happens during transformation. So the last slide is talking about the application of agrobacteria, because the process that I explained before is what happens in nature. However, we can transform the agrobacteria with any kind of gene that we want to transform into the plant. Okay. So this is called transformation, and in nature it happens like the bacteria just infect the plant naturally, but in the lab you have to facilitate that, okay, because it's not as easy, or it will take a lot of time if not. So there are 4 main ways that we can do transformation, and the first one is called stable plant transformation. And it's called stable because in this case the tDNA is always going to be integrated into the chromosome, okay? So, and in one cell, and then this cell is going to generate a whole new plant that is going to be transformed. That means that express this tDNA in its own genome, okay? So that's why it's called stable, because every single cell inside the plant will always have that gene. Okay. And there are two main ways to do a stable transformation. One of them is floral dipping, which is by just literally just putting the plant in a bacteria solution, and that's it. And then the plant eventually produce seeds, and those seeds has plants that are fully transformed. Okay, this is mostly done in Arabidopsis thaliana. And the other way is by doing secondary regeneration, which is what you see in this picture. And this is pretty much the first reason why I got interested in transformation, is because I got to do this process, which is very cool for me. Generating the plants from the tissue is a very interesting thing to do. That's why I started getting interested in that. Okay. However, the main issue with the stable transformation is that this takes a lot of time. It might take between 1 or 2 months to 1 year to get a stable transformant. So of course, if you are in a hurry, that doesn't work. So that's why people generate a faster method for transformation. Okay, and here is when we start with transient gene expression. Transient gene expression, in this case, the tDNA is not. It doesn't have to be integrated into the plant chromosome. That means that you just deliver the tDNA to the plant cell, is expressed, and that's it. Okay, and this is very useful because let's say that you want to test whether your gene of interest has any effect in the plant. Then you can just do transient transformation to see if there is any effect, and if there is an effect, then you can just prove it. And this literally only takes maybe a few days, 3 or 4 days. So that's why it's so convenient, right? The main method that has been established for this is called agroinfiltration. And here I want you to look at this plant leaf, and you see there is a small circle which is yellowish. That means that that yellow circle is an infiltration spot that has been done with a small syringe, and is yellow because the gene that is being expressed is killing the plant cells. Okay, so that means that this person in this experiment, they proved successfully that their gene are making a phenotype in the plant cell. Okay, and you can see that this happens very fast and it's very easy to do, and that's why it's so popular right now. Okay, and then the other two, there is also the method called AgroBest, which is very similar to what I just explained, but in this case they use seedlings. So that means that you don't need to grow the plant until this big. You can just germinate the seeds, and maybe after 10 days you can do the agroinfiltration, and you can see that this plant is expressing something that we call gas, and it's of course it's working. And the last step, which is I would say is the least popular, but it's also very useful for metabolite production. I don't want to go into too much detail, but basically we use the hairy roots producing bacteria that I mentioned at the beginning, and you also express a gene of interest, and in this case it's a fluorescence protein called GFP, and you can see that the hairy roots that are being produced are also expressing the GFP fluorescent protein. So that's how you know that it's working.
Okay, so in this video, Juan present about why he study agrobacteria, and then the importance of agrobacteria, and also how the agrobacteria infect plants, and also mention about the coating, about the secretion system, and then last thing is he conclude the importance of agrobacteria in the application in plant research. So before we end this video, do you have any message for the young people from Myanmar or maybe around the world?
Well, I will imagine it will be to the people from Myanmar, but my message is that if you like science, you should pursue it. And I know it's not easy. I think the most common issue young people that like science is that their parents are like, then if you study this, what can you do with your life, right? So of course the obvious answer for people like me is like, you can get into doing research, but people maybe from Myanmar or from other countries might say, but that's not going to give you any money, or it's not going to be very it's not going to be very exciting in terms of like getting rich or buying a house or getting a stable life. So even if that's true, you can by doing this kind of job, you can also find like yourself useful with your community, and also doing research is a cool thing in terms of like exploring your limitations or the knowledge. And that's one thing that, of course, if you don't like or if that's not fully convincing, that's okay. But the other interesting thing is that you can always go to industry. If you are in, like for example, in this case, there is a lot of, as I mentioned, this gene expression is because it's useful for us. So if you study this, you could easily just go to industry and then get a very good job with very good paid just by doing transformation. And I can tell you that's something that can really happen. So don't think that if you do science you cannot get rich or you cannot get money, because you definitely can. Yeah.
Okay, so thank you so much, Juan, for your time, and then hopefully you can publish more and then have a successful career in your science.
Okay, thank you for watching.